Let's start the chapter 13, that is plant growth and development. You have already studied the organization of a flowering plant in chapter 5. Have you ever thought about where and how the structure like roots, stems, leaves, flowers, fruits and seeds arise, and they too in an orderly sequence? You are by now aware of the terms seed, seedling, plant leaf, mature plant. You have also seen that trees continue to increase in height or girth over a period of time. However, the leaves, flowers and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in a plant? All plant organs are made up of a variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structure and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then, why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote, that is fertilized egg, follows a precise and a highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits and seeds, and eventually they die. The first step in the process of plant growth is seed germination. कौन सी है फर्स्ट स्टेप प्लांट ग्रोथ की? seed germination. The seed germinates when favorable conditions for growth exist in the environment. In the absence of such favorable conditions, the seeds do not germinate and goes into a period of suspended growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. ये जो लाइन्स स्पीड में पढ़ रहा हूँ ये कोई काम की नहीं है, इसलिए मैं स्पीड में पढ़ रहा हूँ इनको, ठीक है। जहाँ पे इम्पोर्टेंट होगी वहाँ पे मैं डबल रिपीट कर दूंगा और स्पीड स्लो हो जाएगी, इसलिए आप डरिए नहीं, ओके। In this chapter, you shall also study some of the factors which govern and control these developmental processes. These factors are both intrinsic internal and extrinsic external to the plant. That is, first, growth. Growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of piece of wood when placed in water? That is imbibition. Am I right? सही आंसर बताया क्या मैंने? How would you describe the swelling of piece of wood when placed in water? That is imbibition. Correct answer है, ठीक है। Plant growth generally is indeterminate. Plant growth is unique because plant retain the capacity for unlimited growth throughout their life. This ability of the plant is due to the presence of meristems at certain locations in their body. The cells of such meristem have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. These forms of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called open form of growth. This form of growth wherein new cells are always being added to the plant body by the activity of the meristem is called the open form of growth. What would happen if the meristem ceased to divide? Does this ever happen? In earlier classes, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of the plants along their axis. You also know that in dicotyledonous plants and gymnosperms, the lateral meristem, vascular cambium and cork cambium appear later in life. These are the meristem that cause the increase in girth of the organ in which they are active. This is known as secondary growth of the plant. कौन से करते हैं? इंक्रीज इन ग्रोथ ऑफ द ऑर्गन में कौन से करते हैं? द लेटरल मेरिस्टेम, वास्कुलर कैम्बियम एंड कॉर्क कैम्बियम, VC, CC और LM. Lateral meristem, vascular cambium, cork cambium appear later in the life. These are the meristem that cause increase in the girth. Understood? हो गया माइंड में सेट? चलो नेक्स्ट is growth. Growth is measurable. Growth at a cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure, one generally measures some quantities which is more or less proportional to it. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. अब protoplasm क्या होता है? Protoplasm is the living material within a cell that is responsible for all living processes. Understood? Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume and cell number. You may find it amazing to know that one single mass root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, growth is expressed as increase in cell number. The latter expresses growth as increase in size of the cell. While the growth of a pollen tube is measured in the term of its length, and an increase in surface area denotes the growth in a dorsiventral leaf. Next is phases of growth. The period of growth is generally divided into 3 phases, namely meristematic, elongation and maturation. Let us understand this by looking at the root tips. The constantly dividing cells, both at the root apex and the shoot apex, represent the meristematic phase of growth. The constantly dividing cells, both at the root apex and shoot apex, represent the meristematic phase of growth. The cells in this rank are rich in protoplasm, possesses large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. The cells proximal just next away from the tip. The cells proximal means pass में just next away from the tip to the meristematic zone represent the phase of elongation. Increased vacuolization, cell enlargement and new cell wall deposition are the characteristics of the cells in this phase. क्या क्या है characteristic? Increased vacuolization, cell enlargement and new cell wall deposition. Further, Away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modification. Most of the tissues and cell types you have studied in earlier classes represent this phase. कौन सा phase? Phase of maturation. ठीक है. Next is growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organism can produce more cells in a variety of ways. ठीक है. डायग्राम दिए हुए हैं. पहला अर्थमेटिक ज्योमेट्रिक ग्रोथ का फेज स्टेज ड्यूरिंग एम्ब्रॉयड डेवलपमेंट शोइंग ज्योमेट्रिक एंड arithmetic phases. So the next is the growth rate shows an increase that may be arithmetic or geometrical. In arithmetic growth, following mitotic cell division, only one daughter cell continue to divide while the other differentiates and matures. In arithmetic growth, following mitotic cell division, only one daughter cell continue to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at a figure 13.5. On plotting the length of an organ against time, a linear curve is obtained. Mathematically, it is expressed at Lt is equal to L0 plus RT, जहाँ पे L0 is equal to length at time 0 और Lt is equal to length at time t. R is equal to growth rate elongation per unit time. Lt का मतलब होता है length at time t. L0 means L0 का मतलब होता है length at time 0. R का मतलब होता है growth rate means और elongation per unit time. Let us now see what happens in geometrical growth. यहाँ पे दे रखा है constant linear growth, a plot of length L against time t. Let us now see what happens in a geometrical growth. In most systems, the initial growth is slow, that is lag phase, and it increases rapidly thereafter at an exponential rate, log or exponential phase. Here, both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of a growth against time, we get a typical sigmoid or S-curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues and organs of a plant. Can you think of more similar examples? What kind of a curve can you accept in a tree showing seasonal activities? सोचो, क्वेश्चन क्या है? What kind of a curve can you expect in a tree showing seasonal activities? A tree that shows seasonal activity will have a sigmoid growth curve, also known as S curve. ठीक है, यही आंसर है उसका. The exponential growth can be expressed as W1 is equal to W0 e की पावर RT. ठीक है, where जहाँ पे W1 is equal to final size, weight, height, number, etc. और W0 means W0 is equal to initial size at the beginning of the period. R is equal to growth rate, T is time of growth, E is base of natural logarithm. Equation है W1 is equal to W0 e की power RT. An idealized sigmoid growth curve typically of cells in culture and many higher plants and plant organs. यहाँ पहले लेग फेस है, फिर एक्सपोनेंशियल फेस है और फिर स्टेशनरी फेस है जो टिपिकल, ठीक है, अंडरस्टूड। पहले है लेग फेस, फिर है एक्सपोनेंशियल फेस और फिर है स्टेशनरी फेस। ग्राफ आपके दिमाग में मैं सेट करता हूँ। चलो, एक्स एक्सिस पे है टाइम, वाई एक्सिस पे है साइज या फिर वेट ऑफ द ऑर्गन। यह है टिपिकल सिग्मोइड कर्व, ठीक है ना, एस कर्व। इक्वेशन एक बार और दिमाग में उतार लीजिए, W1 इज इक्वल टू W0 e की पावर RT, जो कि एक्सपोनेंशियल ग्रोथ की इक्वेशन है। और जो एक और जो इक्वेशन थी पहले वो थी एरिथमेटिक ग्रोथ की, जो LT इज इक्वल टू L0 प्लस RT थी, ठीक है। अब जो चल रहा है अपना W1 इज इक्वल टू W0 ERT चल रहा है, E की पावर RT, ठीक है। हियर R इज द रिलेटिव ग्रोथ रेट एंड इज also the measure of the ability of the plant to produce new plant materials, referred to as efficiency index. Measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence the final size of the W1 depends on the initial size. Final size of the W1 depends on the initial size W0. फाइनल साइज ऑफ द W1 डिपेंड्स ऑन द इनिशियल साइज W0, ठीक है। यहाँ पे एक डायग्रामेटिक कंपैरिजन ऑफ एब्सोल्यूट एंड रिलेटिव ग्रोथ रेट्स। बोथ लीव्स A and B have increased their area by 5 cm square in a given time to produce A1 B1 लीव्स का काम करनी हो, ठीक है। क्वांटिटेटिव comparisons between the growth of living system can also be made in two ways. First, measurement and the comparison of the total growth per unit time is called absolute growth rate. Measurement and the comparison of total growth per unit time is called the absolute growth rate. The growth of the given system per unit time expressed on a common basis. Example, per unit initial parameters is called the relative growth rate. The growth of the given system per unit time expressed on a common basis. Example, per unit initial parameter is called the relative growth rate. In figure 13.72 leaves and we are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A dash and B dash. However, one of them shows much higher relative growth rate. Which one and why? काम करनी है, चलो हटाओ। So the next is conditions of growth. Conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen and nutrients as well as essential elements for growth. The plant cells grow in size by cell enlargement, which in turn requires water. Turgidity of cells helps in the extinction growth. Turgidity of cells helps in the extinction growth. Thus, plant growth and the further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients, macro and micro essential elements are required by plants for the synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases and stages of the growth. Environmental signals such as light and gravity also affect certain phases, stages of growth. Next is differentiation, de-differentiation and redifferentiation. देखो अपन धीरे इसलिए चल रहे हैं ताकि जो है ना लाइंस आपके माइंड में धीरे धीरे गहरी डीप रूट्स में सेट हो जाए, मीन्स आप भूल ना पाओ, ठीक है, ताकि आपको कुछ को रिलेट हो, इसलिए धीरे चलना पड़ा। अंडरस्टूड? सो लेट्स प्रोसीड फर्दर। द सेल्स derived from root apical and shoot apical meristem and the cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. The cells derived from root apical and shoot apical meristem and cambium differentiate and mature to perform a specific function. This act leads to maturation is called differentiation. Clear? During differentiation, cells undergo few to major structural changes, both in their cell wall and the protoplasm. For example, to form a tertiary element, the cell would lose their protoplasm. They also develop a very strong, elastic, lignocellulosic secondary cell walls to carry water to long distances, even under extreme tension. Try to correlate the various anatomical features you encounter in plants to the function they perform. Plants show another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division attained under certain condition. The living differentiated cell that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomenon is termed as de-differentiation. Clear? For example, formation of meristems, interfascicular cambium and cork cambium from fully differentiated parenchyma cells. For example, formation of meristem, that is interfascicular cambium and cork cambium from fully differentiated parenchyma cells. While doing so, such meristem tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific functions, that is, get re-differentiated. List some of the tissues in a woody dicotyledonous plant that are the product of re-differentiation. How would you describe a tumor? What would you call the parenchyma cell that are made to divide under controlled laboratory condition during plant tissue culture? ध्यान से सुनिएगा अगेन. What would you call the parenchyma cell that are made to divide under controlled laboratory condition during plant tissue culture? क्या कहेंगे? Callus cells. ठीक है, parenchyma cells that are made to divide under controlled laboratory condition during plant tissue culture are called callus cells and are an example of de-differentiated cells. Clear? Recall in section 13.1.1 we have mentioned that the growth in plant is open, that is, it can be indeterminate or determinate. Now we may say that even differentiation in plant is open because cells tissues arising out of the same meristem have different structure at maturity. यहाँ पे बन गया assertion और reason. ठीक है, we may say that even differentiation in plant is open. ठीक है, assertion, because reason is tissues arising out of the same meristem have different structure at maturity. That is reason. That is both assertion and reason are correct and reason is the correct explanation of assertion. ठीक है, बन गया assertion reason. The final structure at maturity of a cell tissue is also determined by the location of the cell within. For example, cell positioned away from the root apical meristem differentiate as root cap cells, while those pushed to the peripheral matures as epidermis. Can you add a few more examples of open differentiation correlating the position of a cell to its position in an organ? Next is development. Development is a term that includes all changes that an organism goes through during goes through during its life cycle from germination of the seed to senescence. Diagrammatic representation of the sequence of processes which constitute the developmental of a cell of a higher plant is given in figure 13.8. It is also applicable to tissues organ. चलो ये जो डायग्राम दिया हुआ है सीक्वेंस ऑफ डेवलपमेंटल प्रोसेसेस इन ए प्लांट सेल, इसमें फर्स्ट स्टार्ट हुआ मेरिस्टेमेटिक सेल, ठीक है, वहाँ पे प्लाज्मेटिक ग्रोथ हुई है, फर्स्ट दिया हुआ है सेल डिवीजन, वहाँ पे प्लाज्मेटिक ग्रोथ से मेरिस्टेमेटिक सेल हुआ है, फिर वो प्लाज्मेटिक ग्रोथ होकर डिफरेंशिएशन हुआ है डेथ की जगह, और वहाँ पे एक्सपेंशन, एलोंगेशन, मैचुरेशन, मैच्योर सेल्स एंड सेशन साइकिल दी हुई है, ठीक है। ये इंपॉर्टेंट पैराग्राफ है। प्लांट फॉलो डिफरेंट pathway in response to environment or phases of life to form different kinds of structures. This ability is called plasticity. Example, heterophyly in cotton, coriander and larkspur. In such plant, the leaves of the juvenile plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. This phenomena of heterophyly is an example of plasticity. On the other hand, difference in shapes of the leaf produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. This phenomena of heterophyly is an example of plasticity. Understood? Heterophyly in a larkspur and buttercup. ठीक है, डायग्राम दिया हुआ है। Growth, differentiation and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. The former includes both intracellular genetic or intercellular factors, chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc. Next is plant growth regulators. So starting with characteristics. The plant growth regulators, PGRs, are small, simple molecules of diverse chemical composition. They could be indole compounds, indole 3-acetic acid IAA, adenine derivative N6-4-furfuryl aminopurine kinetin, जो adenine derivative है, or derivative of carotenoids, that is abscisic acid and ABA, और टर्पीन्स गिब्रेलिक एसिड GA3, टर्पीन्स में आते हैं गिब्रेलिक एसिड और GA3, और गैसेस में आती हैं इथिलीन और C2H4. Plant growth regulators are variously described as plant growth substances, plant hormones or phytohormones in literature. The plant growth regulators can be broadly divided into two groups based on their functions in a living plant body. One group of plant growth regulators are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting and seed formation. These are also called plant growth promoters. Example, auxin, gibberellin and cytokinins. These are प्लांट ग्रोथ प्रमोटर्स। प्लांट ग्रोथ प्रमोटर्स के एग्जाम्पल है ऑक्सिन, गिबरेलिन एंड साइटोकाइनेज। अंडरस्टूड? The plant growth regulators of the other group play an important role in plant response to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscissation. The PGR abscisic acid belong to this group. The gaseous plant growth regulator ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. The discovery of plant growth regulators. Interestingly, the discovery of each of the 5 major groups of the plant growth regulators have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptiles of canary grasses responded to unilateral illumination by growing towards the light source, that is phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Figure shows that. In the figure, experiment used to demonstrate that tip of the coleoptile is the source of auxin. क्या है? Tip of coleoptile is the source of auxin. ठीक है? Auxin was isolated by F.W. Wendt from the tips of coleoptiles of oat seedlings. ये important line है. Auxin was isolated by F.W. Wendt from the tip of coleoptiles of oat seedlings. Understood? The Bakane foolish seedling disease of rice seedlings was caused by a fungal pathogen Gibberella fusicoria E. kurosawa. Gibberella fusicoria E. kurosawa in 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with the sterile filtrates of the fungus. The active substance were later identified as gibberellic acid. F. Skog and his co-worker observed that from the internodal segments of the tobacco stems, the callus, a mass of undifferentiated cells, proliferated only if, in addition to auxins, the nutrient medium was supplemented with one of the following: extract of vascular tissues, yeast extract, coconut milk or DNA. Understood? From the internodal segments of tobacco stems, the callus is a mass of undifferentiated cells. ठीक है, यहाँ पर कंफ्यूज करने के लिए डिफरेंशिएटेड आ जाएगा, बट करेक्ट है अनडिफरेंशिएटेड. प्रोलिफरेटेड ओनली इफ इन एडिशन टू ऑक्सीन द न्यूट्रिएंट मीडियम वाज सप्लीमेंटेड विद वन ऑफ द फॉलोइंग: या तो एक्सट्रैक्ट ऑफ वस्कुलर टिश्यूज, या यीस्ट का एक्सट्रैक्ट, या कोकोनट मिल्क और डीएनए. मिलर इट अल in 1955 later identified and crystallized the cytokinesis promoting active substance that they term kinetin. Cytokinesis promoting active substance that they term kinetin. During mid-1960s, 3 independent research reported the purification and chemical characterization of 3 different kinds of inhibitors: inhibitor B, abscission 2 and dormant. Later, all the 3 were proved to be chemically identical. It was named abscisic acid. H. Cousins in 1910 confirmed that the release of a volatile substance from ripened oranges that hastened the ripening of stored unripened bananas later This volatile substance was identified as ethylene, a gaseous plant growth regulator. Let us study some of the physiological effects of these 5 categories of plant growth regulators in the next section. Physiological effects of plant growth regulators. That is first auxin. Auxin from Greek, that is oxygen, to grow, was first isolated from human urine. The term auxin is applied to the indole 3-acetic acid, that is IAA, and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and roots, from where they migrate to the rinds of their action. They are generally produced by the growing apices of the stems and roots. From there, they migrate to the rinds of their action. Auxin like IAA and indole butyric acid IBA have been isolated from plants. कहाँ से आइसोलेटेड हुए हैं? प्लांट्स से. NAA naphthalene acetic acid and 2,4-D 2,4-dichlorophenoxyacetic 2,4-dichlorophenoxyacetic are synthetic auxins. NAA और 2,4-D synthetic auxin है. ठीक है. All these auxins have been used extensively in agriculture and horticulture practices. They help to initiate rootings in stem cuttings, an application widely used for plant propagation. Auxins promote flowerings, example in pineapples. Auxin promotes flowering in pineapples. They help to prevent fruit and leaf drop at early stages but promote the abscission of older mature fruits and leaves, but promotes the abscission of older mature leaves and fruit. In most higher plants, the growing apical bud inhibits the growth of lateral axillary bud. किसकी ग्रोथ को इनहिबिट कर देती है? lateral axillary bud का. Abscission का मतलब होता है the natural detachment of parts of plant, typically dead leaves and ripe fruit. Abscission का हिंदी मीनिंग बताऊं? विच्छेदन मीन्स अलगाव, अलग करना. ठीक है. So the next is, in most higher plants, the growing apical bud inhibit the growth of lateral axillary buds, a phenomenon called apical dominance. Removal of shoot tips, decapacitation, usually results in the growth of lateral buds. It is widely used in tea plantation, hedge making. Can you explain why? It is widely applied in tea plantation and hedge making. Next is, auxin also induce parthenocarpy, example in tomatoes. They are widely used as herbicides. 2,4-D widely used to kill dicotyledonous weeds. Does not affect mature monocotyledonous plants. Does not affect mature monocotyledonous plants. It is used to prepare weed-free lawns by gardeners. Auxin also controls xylem differentiation and helps in cell division. Xylem differentiation में और cell division में कौन help करता है? Auxin. बन गया एक क्वेश्चन. ठीक है. एपिकल डोमिनेंस इन प्लांट एक दे रखा है यहाँ पर डायग्राम. फर्स्ट वाला है ए प्लांट विथ एपिकल बड इनटेक, सेकंड है प्लांट विथ एपिकल बड रिमूव. ग्रोथ दिखाई हुई है. नॉट दैट द ग्रोथ ऑफ लैटरल बर्ड्स इनटू ब्रांचेस आफ्टर डीकैपेसिटेशन. ठीक है. डायग्राम ये रखा है, डायग्राम दिया हुआ है. सो द नेक्स्ट इज गिबरेलिन्स. Gibberellins are another kind of promotory plant growth regulator. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellin to be discovered and remains the most intensively studied form. GA3 was the first gibberellin, ठीक है, to be discovered. All gibberellic acids are acidic. ये assertion बन सकता है और ट्रू और फॉल्स में भी आ सकता है स्टेटमेंट. All gibberellic acids are acidic. That is true. They produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of grapes stalks. Gibberellins causes fruit-like apple to elongate and improve its shape. They also delay senescence. एप्पल को कैसे हेल्प करता है? एलोंगेट और शेप को इम्प्रूव करने में, इम्प्रूव इट शेप्स, ठीक है. दे ऑल्सो डिले सेनेसेंस. सेनेसेंस का मतलब क्या होता है? द प्रोसेस ऑफ बायोलॉजिकल एजिंग वेयर सेल्स स्टॉप डिवाइडिंग एंड एज परमानेंटली विदाउट टाइम. समझ गए? हिंदी में मतलब बताऊँ तो बुढ़ापा. Thus the fruits can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugarcane stores carbohydrate as sugar in their stems. Sugarcane stores carbohydrate as sugars in their stems. As sugar in their stem में store करते हैं, ठीक है. Spraying sugarcane crop with gibberellins increases the length of the stem, thus increasing the yield by as much as 20 tons per acre. 20 tons per acre. How many tons per acre? 20 tons per acre. ठीक है. चलो तो next proceed further. Spraying juvenile conifers with gibberellic acid GAS hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting, that is internode elongation, just prior to flowering in beet, cabbage and many plants with rosette habit. ठीक है. किन किन में? बीट में, कैबेज में और many plant with rosette habits. Next is cytokinins. Cytokinins have specific effects on cytokinesis and were discovered as kinetin. Cytokinins have a specific effect on cytokinesis and were discovered as kinetin, a modified form of adenine, a purine. Kinetin is a modified form of adenine, a purine, from the autoclaved herring sperm DNA. From the autoclaved herring sperm DNA were discovered as kinetin from the autoclaved herring sperm DNA. ठीक है. Kinetin does not occur naturally in plants. Search for natural substance with cytokinin-like activities led to the isolation of G18 from the corn kernels and coconut milk. From the corn kernels and coconut milk. Since the discovery of G18, several naturally occurring cytokines and some synthetic compounds with cell divisions promoting activity have been identified. Discovery of G18, several naturally occurring cytokines and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in rains where rapid cell division occurs. कहाँ पे होते हैं? किस राइन में? Where rapid cell divisions occurs. For example, root apices, developing shoot buds, young fruits, etc. Repeating it again. For example, root apices, developing shoot buds, young fruits, etc. सेट हो गया माइंड में? चलो नेक्स्ट. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth and advantageous shoot formation. किस किस में हेल्प करता है? हेल्प टू प्रोड्यूस न्यू लीव्स, क्लोरोप्लास्ट इन लीव्स, lateral shoot growth and advantageous shoot formation. Understood? Cytokinins helps overcome the apical dominance. Cytokinin किस में हेल्प करता है? एपिकल डोमिनेंस को ओवरकम करने में. ठीक है. दे प्रमोट न्यूट्रिएंट मोबिलाइजेशन विच हेल्प्स इन द डिले ऑफ leaf senescence. They promote nutrient mobilization which helps in the delay of leaf senescence. So the next is ethylene. Ethylene is a simple gaseous plant growth regulator. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis and apical hook formation in dicot seedlings. ध्यान से सुनना. Influence of ethylene on plant include horizontal growth of seedlings, swelling of the axis and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plants organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in rate of respiration is called respiratory climatic. Ethylene breaks seed dormancy and bud dormancy. Initiate germination in peanut seeds, sprouting of potato tubers. हो रहा है धीरे धीरे माइंड में सेट, इसलिए स्लो पढ़ रहा हूँ मैं. ठीक है. सो द नेक्स्ट इज इथिलीन प्रमोट्स रैपिड इंटरनोड petiole elongation in deep water rice plants. It helps leaves upper part of the shoot to remain above the water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used plant growth regulator in agriculture. The most widely used compound, a source of ethylene, is ethafone. Ethafone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Slowly की जगह fastly कर सकता है, but correct है slowly. Understood? Ethafone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut. Again repeating it. Ethafone hastens fruit ripening in tomatoes and apples and accelerate abscission in flowers and fruits. Thinning of cotton, cherry, walnut. It promotes female flower in cucumbers, thereby increasing the yield. क्या प्रमोट करता है? फीमेल फ्लावर्स इन क्यूकम्बर में, जो किसमें हेल्प करता है? इंक्रीजिंग द यील्ड. सो द नेक्स्ट इज abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy. But like other plant growth regulator, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. Abscisic acid inhibits seed germination. Abscisic acid stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore, it is also called the stress hormone, which is a very famous question. Abscisic acid ABBA plays an important role in seed development, maturation and dormancy. किसमें important role play करता है? First, seed development, maturation and dormancy. By inducing dormancy, abscisic acid helps seed to withstand decision, dissection and other factors unfavorable for growth. In most situations, ABBA acts as an antagonist to GA. Abscisic acid acts as an antagonist to gibberellic acids. We may summarize that for any and every phase of growth, differentiation and development of plants, one or the other plant growth regulator has some role to play. Such roles could be complementary or antagonist. Such roles could be complementary or antagonistic. This could be individualistic or synergistic. Similarly, there are number of events in the life of a plant where more than one plant growth regulator interact to affect that events. Example, dormancy in seed, buds, abscission, sensation, apical dominance, etc. When we summarize that for any and every phase of growth, differentiation and development of plant, one of the other plant growth regulator has some role to play. There are a number of events in life of a plant where more than one plant growth regulator interact to affect that event. Example, dormancy in seeds/buds, abscission, sensation, apical dominance, etc. Remember, the role of plant growth regulator is of only one kind of intrinsic control. Along with genomic control and extrinsic factors, they play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via plant growth regulator. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature. Both of them the extrinsic factors on initiation of flowering. ठीक है? So this is the summary. We are going to read in a speedy way. Growth is one of the most conspicuous event in any living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involves increased protoplasmic material. In plants, meristems are the site of growth. Root and shoot apical meristems, sometimes along with intercalary meristem, contribute to the elongation growth of plant axis. Growth is indeterminate in higher plants. Following cell division in root and shoot apical meristem cells, the growth could be arithmetic or geometrical. Growth may not be and generally is not sustained at a high rate throughout the life of a cell, tissue, organ, organism. One can define three principal phases of growth: the leg, the log, and the senescent phase. Leg, log, and senescent phase. When a cell loses the capacity to divide, it leads to differentiation. ठीक है? पहले क्या करेगा? Differentiation. फिर differentiation results in the development of a structure that is commensurate with the function of cells finally has to perform. General principles for differentiation for cell, tissue, and organ are similar. A differentiated cell may de-differentiate and then re-differentiate. पहले होगा differentiation और फिर differentiated cell क्या कर सकता है? मैं de-differentiate and then re-differentiate. Since differentiation in plants is open, the development could also be flexible. That is, the development is the sum of growth and differentiation. Development is the sum of growth and differentiation. Plant exhibit plasticity in development. Plant exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factors. Intercellular intrinsic factor are the chemical substance called plant growth regulators. There are diverse groups of plant growth regulator in plants, principally belonging to 5 groups: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. Auxin, gibberellin, cytokinin, abscisic acid, and ethylene. These plant growth regulators are synthesized in various parts of the plant. They control different differentiation and developmental events. Any plant growth regulator has diverse physiological effects on plants. Diverse plant growth regulator also manifest similar effects. Plant growth regulator may be synergistically or antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. With this, we completed the chapter. Thank you for hearing.